Have you ever wondered how arsenic affects aquatic life, particularly fish? Well, arsenic, a toxicant found in freshwater habitats, can have some seriously damaging effects on our finned friends. Today we'll be looking at the fish species known as Cirrhinus Mrigala. This study's aim is to evaluate the lethal toxicity of arsenic trioxide on this species over varying exposure durations. With different exposure times and the analysis of various serum biochemical parameters, we are about to dive deeper into this unique study. To unravel the mystery of arsenic's impact, a study was conducted on Cirrhinus Mrigala, exposing them to arsenic for 24, 48, 72, and 96 hours. The objective? To understand the lethal toxicity of arsenic trioxide on this freshwater species. The study was meticulously designed, closely observing specific parameters over varying exposure durations. These parameters encompassed vital elements found in the fish's bloodstream: sodium, potassium, chlorine, urea, protein, glucose, albumin, alanine aminotransferase, and aspartate aminotransferase. Each parameter was carefully measured and recorded, providing a holistic view of the arsenic's effects. The fish were divided into two groups for a comprehensive comparison. One group served as the control, experiencing a normal environment, while the other was subjected to the arsenic exposure. The biochemistry of these two groups was then compared, painting a clear picture of the changes induced by arsenic. These parameters were analyzed in both control and arsenic-exposed groups, revealing significant variations. What did the results show about the arsenic's impact on aquatic life? Well, the findings were quite revealing, to say the least. The study unveiled some notable variations in the serum biochemical parameters of Cirrhinus Mrigala under acute exposure to arsenic trioxide. Interestingly, the results showed a decrease in certain parameters. The levels of sodium, chloride, and albumin were recorded to be lower in all arsenic-exposed fish groups compared to the control group. This implies that arsenic exposure might disrupt the balance of these essential elements in the fish's system. On the other hand, it wasn't all about decline. Some parameters showed an increase under arsenic exposure. Potassium, total protein, urea, total glucose, as well as alanine aminotransferase and aspartate aminotransferase were all recorded higher in arsenic-exposed fish groups. This indicates that arsenic might stimulate the overproduction of these substances, causing a detrimental effect on the fish's health. So, what does this all mean? The variations depicted in the serum biochemical parameters under arsenic exposure suggest a disruption in the fish's internal system. The results clearly indicate arsenic trioxide's adverse effects on these parameters. This brings us to an important conclusion. These serum constituents could serve as bioindicators to assess the toxicity caused by arsenic. They provide critical clues to the health status of fish, thereby playing an important role in environmental monitoring. The findings clearly indicate arsenic trioxide's adverse effects on serum biochemical parameters, suggesting that these constituents could serve as bioindicators to assess the toxicity caused by arsenic and thus help in assessing the health status of fish.